or anytime you want to access the fruit. So they made a slightly more intensive strategy of where you support the netting off of the orchard in what's called a driving enclosure. You use trellis poles and metal wires, hangs it up off the trees, that way the control drive tractors, then you can access the fruit for pruning it, harvesting without having to take it down. Basically, the only time you have to take it down is the top for winters if it snows. And here in Washington, this type of netting of driving enclosure has become a lot more common since 2015 when I started in grad school. And that's kind of why my project is focused on it, to see what is this new full enclosure doing to apple insect communities, since it hasn't been studied as well as the other structures. Now these full driving enclosures have a lot of other functions as well. So they also reduce heat stress to the tree and to workers. A lot of people have mentioned that working under this netting is actually preferable because they're now experiencing less sunlight directly shaded for them. It eliminates the need for that overhead cooling, which can cause secondary issues of like diseases such as mildew because you're adding all that water into the canopy. It's been shown to improve fruit skin size and color. Sorry, fruit skin quality, size, and color. It reduces worker stress in general, and the equipment can still access it. Since it is a full enclosure, it can keep out birds. This is the main structure that's been shown to reduce wind, and it can exclude deer. But one of the big questions is what does it do to insects? And as an entomologist, that's the part that I'm really focused on. So, as any good researcher, we just decided to go to a research plot and build small-scale small-scale style cages that represent these larger commercial block orchards. And we have a small cage, that's a really small scale, which only covers 3 trees. Here's 10 feet tall, 10 feet wide, 5 feet deep, and larger scale, which cover 48 trees. These are all done at our WSU Research Sunrise Farm down by Rock Island, which I'm sure a lot of you have been to. And we tested 3 treatments. We wanted to see, does a cage over a conventionally managed orchard enhance control of pests and cause non-target effects to secondary pests and their natural enemies, worse than a conventional orchard does by itself, or compared to a control where there's no treatment at all. So, one of the primary pests we looked at was Codling moth. Codling moth, key pest of apple. In this research block, it is very abundant. It can cause up to 100% loss every year if it's not treated. I'm sure our orchard manager Cameron would attest to that if he was here. So luckily we have the key pest there. It's abundant and it's easy to test for. We just built a cage, put up some traps to monitor it throughout the year, and assess fruit damage each year. These trials were done from 2016 to 2017, and since they were very similar, later on when I show the results, I'll show the results for both the small cage and the large cage side by side. They were basically identical, just difference of scale. For the non-targets that we want to look at, we want to see does this netting exclude beneficial insects and cause secondary pest flare-ups. One of the biggest secondary pests that we have are aphids on apple trees, and here we looked at woolly apple aphid and its natural enemy complex, which consists of circuit flies, lacewings, its parasitoid Aphelinus molly, which if you're not familiar with what a parasitoid is, it's a lot like the movie Alien, where it lays an egg inside its host. Egg develops into larvae, eats the host, pops out as an adult, goes and does it again. In this case, it's a wasp. So it's minute and harmless to us, but a great enemy of aphids. And then if you were here for Robert's defense, you could learn how earwigs were great beneficial insects to control woolly apple aphids, so we also monitored those. For monitoring insects, it's usually not too labor intensive when there are traps built for them. Luckily, there were traps for almost each insect we looked at, except for woolly apple aphids, we just did time counts. So for earwigs, we put out cardboard domiciles, 1 per plot. We collected them once every 2 weeks, just to count the amount of earwigs that were out there. After opening them up, we released all the earwigs back into the plot to not remove them. For the little parasitoid Aphelinus molly, it's attracted to yellow, so we put the little yellow sticky cards out, brought those in once every 2 weeks, counted all of them on it. And then this here is a white panel trap with a plant volatile lure that attracts both lacewings and circuit flies, and was actually developed by Vince Jones over in the other lab, at least in part by Vince Jones, so I can thank him for that. And so basically we trapped for all species except for woolly apple aphids, which we counted once every 2 weeks, and we accumulated the counts throughout the season to see was there a difference in overall abundance throughout the year for each pest or natural enemy. What did we find? So like I said earlier, I'll have the small cages here and the large cages here, representing the same species. In this case, it's Aphelinus molly. which I provided a picture of the insects, or yeah, basically all insects for your reference. This is a parasitoid apheline small laying an egg in a woolly applet, but very zoomed in. Thanks to Betsy Beers, it's a great picture. On these graphs on the y-axis, we have the cumulative counts of the amount of insects that we caught. And on the x-axis are our 3 treatments. So we have the cage, air blast is the conventionally managed plot, and then the check, which is no treatment at all. As you can see, for both years in the cage, 2016, there's a lot more apheline molly than the other trees, and in 2017, there's still a significant amount. So we have a lot of parasitoid present in these cages. Earwigs, more good news is we're not knocking out the earwigs. In 2016, we had more actually in the cages than 2017. Similar amounts for 2017 and similar amounts in the large cages for both years. So we're not losing our parasitoid or a great beneficial predator, which is good. However, for our alate, bigger predators that fly around, we are knocking those down quite significantly. In the cage, we had almost no lacewings lacewings either year. By the end of the year, we were actually just getting zeros, basically suggesting that we trapped them out and that they never were able to reestablish. And same news for surfer flies. We only caught 1 that year in 2017 in the small cages, and then 2016, and basically none as well in the large cages for both years, whereas they were prevalent outside the cages, showing basically that netting is not letting them in, and our traps took out any existing population quickly. So we have parasitoid, 1 generalist predator, but we're missing 2 other very beneficial predators. And when that happens, you're kind of disrupting biological control. And go figure, when you look at your aphid pest, who is now missing 2 predators, its populations just soar. Take it. This is a break in the axis because these numbers are under 100 colonies, whereas these are upwards of almost 10,000 colonies. Again, this is cumulative. So I wasn't out there counting 10,000 each week. But it still shows a good picture that the woolly Appalachians were very much so infesting these cages. I have many coats that are just covered in them that I have used as protective shield from their wool, compared to where their predators are present. So we believe that the lack of those predators is what caused this outbreak, because there are no other differences between the treatments. But enough about the secondary pests, what about the primary one? Codling moth. So good news, codling moth, which is a horrible pest of apples. Real quick, does anyone know what's worse than finding a caterpillar in an apple? Finding half a caterpillar. Yeah, gross. So good news, cages were reducing significantly the amount of codling moth that we were capturing inside. So y-axis is cumulative codling moth adults that we caught both years. For the small cages, since it was such a small plot, we didn't have enough distance between the reps to test the air blast treatment as well, so we just did the gauge versus control, whereas the larger blocks were easier to separate out, reducing the chance of basically interference between replicants. But in both trials, cages caught significantly less codling moth. That's good news. However, they weren't gone completely, so that is something I'll get into in a little bit. As you can guess, when you have less adult density, you have less damage. And the surprising thing here is that this was a conventionally managed block, which should demonstrate exactly how much pressure these blocks had, because usually that knocks it down to like 1% or less of damage, and we were still upwards of almost 5, just because these blocks are infested with codling moth. The control, sorry, I should add, this is first generation damage. This isn't even before they made it the end of the season. This is about like June. The control is already at 20% damage. And the cage is right on track, way down below, showing that significant drop, even above conventional. This trend held true all the way through the pre-harvest, where the control shot up to about 90% damage. The conventional stayed about 5 to 10, and the cage was significantly below that, below 5. But we didn't completely knock out codling moth. So there's a theory out there that this netting is a physical barrier preventing them from getting in. Now, when we built these cages, we built them over an existing orchard. So there could have been an internal population of codling moth that we built these cages over, and that might be what we were catching, or it could be that they are able to get through the netting, and we just can't tell. So I wanted to go into that for 2018 and 2019 to see, is this netting actually permeable to codling moth or not? So to start off, we used only the large cages for this. we had 2 treatments, cage control, to see is the netting blocking them, and we had to get rid of the internal population. So in 2018, we sanitized the cages. For those of you who worked here for a while, that's Bruce. He's retired now, but he helped me build all of these, and that was a fun experience because it gets windy, and those things are big kites, basically. But for 2018, 2019, we kept them up, and we kept them completely sealed with a lot of effort, and we decided to try to get rid of the collie moth population. You should get rid of its host, which is apples. So we went through in June, removed all of the fruit in every cage and every control plot. We wrapped cardboard around the trees because as a collie moth matures, as it's about to turn to an adult, it crawls down usually towards the soil or it nestles within the bark of a tree, pupates, then pops out as a moth a little bit later. And if you put this cardboard around a tree, it'll pupate inside there, and you can remove it. Remove any collie moth that were coming off that tree. So we did that for every generation, and by good scientists, to make sure that what we were doing was working, we tracked the collie moth populations in the cages through traps and by counting the bands each year. So this graph here on the y-axis is the number of adult collie moth that we caught. Treatments are on the x-axis, and red, or on the left if you can't tell, is the amount that we caught before we removed the apple, and black is the amount that we caught after we removed the apples. So within the cage, there were quite a bit before we removed the fruit. That dropped down to almost 0 after we removed it. That's the rest of the season from June through October. Whereas in the control, we still caught quite a bit after we removed fruit, likely coming from the other plots. So this suggests that the netting should maybe be a barrier because now we've almost eliminated collie moth in the plot and our trap capture went down. To further show that, we looked at collie moth larvae. During the first generation, when there was fruit, so this is right before we removed it, there were a lot of larvae in the cage and in the check. After we removed the fruit, those dropped down to 0, which shows that biology is holding true. When you get rid of the food, they can't survive, and we're getting rid of all the population. So this is for 2018. So going into 2019, we can pretty safely assume that we've knocked down the population a lot, if not to nothing. So in 2019, we want to see, were they able to reestablish? If we put traps in those cage plots, will we catch more collie moth, showing them that they are getting through the net? And we trapped from March through September, which is just the general time of collie moth flight. And here on the y-axis is the average amount of collie moth we caught per plot. The x-axis is time. The red line is the amount that we caught in the cage, and the black line is the amount that we caught in the control. So collie moth had 3 generations that year. As you can see in the first generation, we caught a lot in the control. We caught about 1/9 of that within the cage. By the second generation, there was no difference between the 2 treatments. If you look at it as a cumulative count, this is in the cage and the control. We caught about half as many within the cage throughout the season as the control, showing that it was significantly reducing it, but it did not prevent it completely. Looking at damage, we had about the same thing at the first generation when there was that big difference in the amount that we caught. There's also a big difference in damage. So first generation so far, netting is looking pretty good. It's not perfect. But by the end of the year, when we get to pre-harvest, there is no difference in damage. Both of them are up in the 90% because there's no insecticides to protect that fruit. And the collie moth are in there in great abundance. But what is this telling us? Is this telling us that it did prevent some in the beginning, but then it became a really leaky cage and all of the other collie moths just kind of flooded in? Or is it more of a testament to that it only takes a few collie moth to catch up to population? And so if you don't control this little bit, you're going to end up with no control later on. And that is a pretty striking difference, showing that the netting should at least be acting like some of a barrier because it prevented a lot from going in there. So to get that further, we want to see if we can control how many moths there are, we know, can we tell how many are getting in or out of the netting? And for that trial, this is also 2018 and 2019, I used the small cages, and we went up to Canada and got some sterile moths, which are irradiated to make sure they can't reproduce, and they're marked with a dye on the inside so you can identify them as sterile. But to take it even further, we marked them with fluorescent powder as well. So we marked a bunch of sterile moths with powder, took them to the salon for a day, made them look nice, and we released them inside the plots and outside the plots to see are moths escaping the cage and can they get into the cage at the same rate as the net completely permeable. And then we took them back to the lab, shined UV light on them, put it on some rave music, had a good party, looked at all the colors that we could see. So just to get terminology correct, when we release moths outside and try to recapture them inside, that's immigration. They're immigrating into the cage. Got something fun there. When we release them inside the cage and catch them outside, they're emigrating. So emigrating into, emigrating out of. We want to see, are those rates the same? So for 2018-2019 moth immigration, we're out of the plot. We released them inside, put traps all around each plot to try to catch them. What do we find? Well, in 2018, for the cage versus the control, here on the x-axis, cage control, y is the amount that we caught. You can see that the cage caught a lot, significantly less than the control did, showing that maybe the netting was preventing some of them from getting out, but some still didn't. And in 2019, that difference disappeared. There was lower capture overall here, maybe due to weather, maybe due to sprinklers. We don't know what was going on that week, but that difference is gone. It's showing that the netting, basically, if they're flying out of the netting, they can get through it. However, when they're going into the netted plots, it's a very different story. In the control, we had high recapture both years, and in the cage, we caught almost nothing. There's only one moth here and 0 moth there. So even though they can get out of the cage, showing the net is permeable, for some reason they're not flying into it. And we're thinking this is more of a behavioral avoidance, or rather than a physical barrier. It's just they don't want to be under that netting. It's disrupting lighting. It's a tight enclosure. It should be replicated on a bigger scale to see if that holds true, because maybe it was just that small 3-tree area they don't want to be in. But it is interesting that it had such a difference between which way they were moving. So overall, what did we find through shade net enclosures was that full enclosures did significantly enhance colony moth control when added to a conventional plot. And when you have netting alone with no other treatments, it did not prevent damage throughout the year. So it is maybe not a standalone tactic, but a good tool to add into the overall management tactics. Only a few colony moth are needed to start that big population, which we've known that for a while. Colony moth are very dependent. And always with everything, you're trying to pay attention to natural enemies and the secondary pests that they control, because when we knocked out 2 of them, we did have huge flare-ups of void applications. So those are my full cage trials. To move into another thing, since I was looking at netting and how it affects all insects, we wanted to see what does netting, is there a way we can help growers who are trying to deal with stink bugs with the idea of physical exclusion. So in Washington State, there are over 50 species of stink bugs. That's a lot, but luckily, here in central Washington, there are really only 3 main species that are of agricultural concern right now. And that is the red-shouldered stink bug, which is about a ½ inch. The conchuela bug, which a lot of people just call the crow stink bug. This is a conglomerate of 4 species that are very hard to identify. Differences between the species, so you just kind of call them all crow. And they're the biggest one, they're about 1.5 inches. And then the consperse, which is right in the middle, is about 1 inch. So those are the 3 main species that most growers have to worry about every year for now. And they're all a single generation per year, unlike codling moth, which have 3. They come out as adults from overwintering in late April or early May. They start laying eggs out on the natural native vegetation that exists. They really like our shrub steppe area. So they lay eggs for a couple weeks. Those eggs begin to hatch. They go through 5 nymphal instars from June through August. Around late August, they start becoming adults again. And then they live as adults all the way until the next year, overwintering, to start the cycle over. Now, for most of the year, they live out on that native vegetation I was talking about. They live out on bitterbrush and currant and lupins, because these are all seed-bearing structures, seed-bearing plants that have protein-filled structures like this that they love to feed on. But, kind of like I was talking about their arid climate and sunburn, these plants don't live throughout the entire year. They dry out in senesce and end up dying, and they're no longer a viable host for these insects. And it's believed that when this happens, they migrate, stink bugs will move from these non-viable hosts into an orchard, which at this point is green and lush and starting to get fruit, and that's when the growers experience their damage. So it's been suggested from people who have done observational studies on stink bugs that they climb up any vertical surface they find, which you can experience anytime you want if you come by the lab, and they will fly off of that point and kind of remain at that height. all the way into the orchard. So people have found that when they put up physical barriers by cotton from corn, they can prevent it from going into the cotton because they're flying at the height of the corn. However, if they put a physical barrier up next to a giant woodlot that has like 40-foot trees, they did not experience the same block because they're flying over it. So since we have shrub step vegetation that kind of lives or grows to roughly 9-10 feet at max, the idea of a physical barrier might actually do something. And it's coming into a very critical time because currently sampling for stink bugs is difficult. They are hard to track. We have traps that use aggregation pheromones. Unlike sex pheromones that we use for codling moth, it just kind of brings them into the area. They're not going to go inside the trap. So you go to the trap, you may not see any stink bugs, but if you look around the area, you can find more of them. We also have people like this who dress up in our fun hats and pose for fun pictures. And you go around to plants and you hit it with a stick and stink bugs come flying out onto your sheet, then you know they're there. If they don't, you don't know if you hit the wrong branch or if they're just not there. So in general, it's hard to tell where the stink bugs are and when. And this has made management difficult because they believe that it's when those plants die that they get all their stink bug pressure. And one of the only responses they have right now is to spray broad spectrum insecticides because stink bugs are pretty resilient buggers. And since we work with apples, any cosmetic damage is still damage, and we need to prevent that. And just like with barriers, when you knock out all the beneficial insects, you get secondary pest outbreaks like mites and woolly apple aphids. And the biggest thing that I triggered why I wanted to research this in the beginning was growers were still telling me they were getting damaged just by doing this. So they think they're spraying at the right time because that's when the stink bugs are there. They're getting these secondary pest outbreaks, which isn't good, and they're still getting damaged. So none of it sounds great. Luckily, there has been some research to try to help prevent the secondary pest outbreaks from the sprays. Stink bugs, since they migrate into orchards, usually occur around the border of the orchard first before going inside. So the idea was that if you just spray the border rows, you'll kill most of the stink bugs that have moved into the orchard, but save about 90% of the orchard from being sprayed, creating a refuge for all the beneficial insects. To take it a step further, they found that if you attach one of those pheromones that attract them to a tree, you can bring all the stink bugs in this area to that tree or the ones right next to it, and just spray those 3 trees. And that reduces spray about 99%, basically eliminating the whole secondary pest outbreak thing because you're not killing all the beneficials. But I want to see if we could just prevent them from ever getting in the orchard, then we would never need to spray for them anyways. And since all this netting is going up everywhere, just building a single wall instead of a giant cage would be a little easier, and it might be able to prevent them from flying in. And to do this, we had to figure out when are stink bugs flying in, how high are they flying in, and then test a physical barrier and can it stop them. So, to determine when they flew in, I met a guy who makes giant clear sticky traps. I was like, that's perfect, because I don't want them seeing the trap when they're flying into the orchard. And we built these huge, giant, clear sticky kites, like I like to call them, because the first year we built them that were 6 feet by 9 feet, did not bury the post deep enough, so I came back to the orchard, they were gone completely. And I was like, well, where'd they go? So I walked around, and they covered the top of the trees. They'd flown 15, 20 feet, and I wish I could have been there to see that. That would have been amazing. But realized 6 by 9 feet wasn't going to work because in 2017 we put them back. We didn't find, basically the stink bugs were flying higher than 9 feet, so the next year we built them higher, up to 13 feet, and we did that for 2 years. Checked them throughout the summer, every week, when are stink bugs flying in versus out of the orchard, and at what height are they flying. What did we find? Well, on this graph, on the y-axis is the number of stink bugs we caught per barrier, x-axis is the dates that we were checking it. The red line is the stink bugs that were flying from the vegetation into the orchard. Black line, stink bugs flying back from the orchard into the vegetation. And as you can see, here in early June in 2018, we're already getting stink bugs flying into the orchard. Most of them flew in around the middle of July, and the interesting part was that once they flew into the orchard, they actually flew back out. It wasn't just a one-way migration, they were kind of going back and forth throughout the summer. And then by August, it kind of tapered off, and the migration kind of slowed down, likely because at that time is when all the plants are dying. There's no reason to move back out. So, talking to growers, one of the hypothetical spray dates that they usually go by is early August. So let's say that was there. if all these stink bugs flying into the orchard at this time were not being affected by that spray there, it would kind of explain why they were still seeing damage. So that was kind of an important thing to find out. And like all good science, you want to see, does that hold up each year? So red line into the orchard, black line back out into the vegetation, hypothetical spray date. Sure enough, there's still a lot of area of stink bugs being in that orchard, not being treated. It might be why they're still getting a lot of damage. So what about height? So we know timing now. How high are they flying? This is them flying out of the orchard back into the vegetation. So y-axis is number of stink bugs, x-axis is height, in feet and meters for anyone who is not in the US, so might say meters. From the orchard, you can see there's no real difference. They're kind of all flying at various heights, which we expected because trees are a little bit taller. Our barriers didn't go up above the trees. Stink bugs climb up to whatever branch they reach, fly off. Not much difference. But we don't really care how high they're flying out of the orchard. We care how high they're flying in. And luckily for us, we predicted it a little bit better. And there's kind of a bell-shaped curve here, where at the higher heights there's less stink bugs, at the lower heights there's less stink bugs, with most of them flying between about 1 and 3 meters, which is 3 to 10 feet for everyone else in 2018. And in 2019 that held true as well. See here at 11 and 12 feet there's almost nothing, and at 0 feet, or 0 to 1 foot there's really nothing. And between 3 and 8 there's still that majority of stink bugs flying in. So an 8-foot barrier doesn't sound too hard to do considering those cages we were building were 15 feet. But you don't want to stop at 8 feet just in case there are a few flying over it. So we built giant 12-foot barriers by 150 feet here, because in the Beers Lab we like to make dreams a reality. Nothing is too big. And also because I needed to work on my ability to sew, we decided to put these little flaps in the barriers here to capitalize on that behavior, that when they climb up, they're going to reach whatever they can at the highest point. When they reach that flap, they'll turn upside down and they'll stop moving or fly back off into the vegetation, and that will prevent them from crawling over and then flying into the orchard. So we had 3 of these that were 150 feet long, and we built these in 2016, did a couple tests with them, decided we need to step up our game, and they made a black netting that has insecticide infused into it, and we put that in the flaps of half the barrier, so that way we would have 3 treatments, one with barrier with insecticide flaps, barrier with normal flaps, and then a no barrier control that you can't see over here. There's nothing there. The idea here was that they would fly up, crawl up into that flap, get treated with the insecticide, die, preventing them from ever either reproducing or going around the barrier or going over. And we tested this for 2 years, from 2018 to 2019, growing season from June to September. So this is what one of the sites looked like up in Manson, to tell you what I was doing. Here's the barrier, here's the control. Barrier is cut in half. This half had black insecticide netting, this is just normal netting. And each week we would go out and we would sample the orchard. Here we are again doing our fun stuff. Sampling the orchard for stink bugs. We would beat sheet the trees, doing 10 trees in each area. And each tree we would hit 4 times to kind of look all over that we basically everywhere that we could reach to try to catch any stink bugs. Accumulated all the stink bugs we caught throughout the year for each treatment in the orchard. We then go out into the vegetation to do the same thing to show that there were stink bugs there, because if you went out and didn't find any, that'd be a bad place to do a research site. Basically the same thing we did. Instead of doing 8 trees, we did 8 minutes to try to keep it the same, and we beat sheeted the vegetation to catch all the stink bugs that we could. And then, because we always care about non-targets, we put out these these brown tarps underneath the netting to see, is that insecticide netting also killing beneficials that are trying to fly into the orchard. And because of good science, we had to put one in each place. So this one was just a tarp out in the middle of the field. So from our vegetation samples and orchard samples, we kind of track the adults and nymph cycle of stink bugs and when they are present. Here on the y-axis is the number of stink bugs we caught, x-axis is dates. The gray line is nymphs, so this just shows that there's still really only one generation of stink bugs. The black line is adults out in the vegetation, and the red line is in the orchard. So by this, if we just had beat sheeting samples, we would still show that adult stink bugs really aren't working in the orchard until August, and that's probably why they thought this for so long, because that's just what beat sheeting takes a lot of population to find it. When you accumulate it all, This is for 2018, so this is the cumulative stink bugs in our 3 treatments. Red is the amount that we caught in the vegetation, black is the amount that we caught in the orchard. As you can see in the vegetation behind the netting, there was significantly more stink bugs than behind the control. However, there was no difference in what we found in the orchard. So if you look at it this way, it kind of seems like the netting didn't really do anything to stop stink bugs in the orchard. But another way to look at it is that the netting actually prevented about 86% of the stink bugs from making it into the orchard, compared to where if there was no net, it'd be about 40%. So this was suggesting to us that the netting was actually blocking a lot of them from making it into the orchard in 2018. Fortunately, in 2019, that trend did not hold up because we caught a lot more in the control section, and the orchard still was not significantly different, vegetation was still not significantly different, and now the amount making it into the orchard wasn't different. So another hypothesis is that these orchards are commercial orchards, and the growers are still spraying. So maybe the reason we're catching so little adults in the orchard is because they're spraying, and it's hard to see what the netting was really doing. So it would be good to redo this trial on a research farm that had a high stink bug density to see is the netting actually blocking. But the other fun part that we got to do was look at non-targets. So here's one of those tarps, and here's the netting. We just put the tarp right up against the netting to catch anything that fell. And if any of you have ever been just tromping around in the vegetation of central Washington, you know that insects are not the only thing that's out there. You actually find a lot of fun things. This was a pretty cool guy that I caught. He was running around. Luckily, was not hurt, completely alive. Another fun one that I'm sure you guys are all aware of are these ones. They like to hiss and snap a little bit, but if you hold them just right, you can carry them off and make sure that they don't hurt you. So I found a lot of rattlesnakes. One time I was crawling onto the tarp to pick up stink bugs and other insects, and I put my hands down, and they both just started vibrating. And I was like, oh, insects don't do that. That can't be right. So flipped up the tarp, sure enough, there were a couple rattlesnakes, moved them off, everything was fine, went back and caught the stink bugs. No snakes were harmed in any of these experiments. But back to entomology. So we looked at what things was this netting killing, including stink bugs and non-targets. So here, y-axis is the total cumulative capture on the tarps of whatever insect we're looking at. In this case, it's Orthoptera. Orthoptera are grasshoppers, and I included a picture of each insect in the upper corner for those of you just not familiar with them. We had our 3 treatments and 2 years. So the deltamethrin killed a lot more grasshoppers than the full netting by itself, which killed even more than the control. We did catch one grasshopper on a tarp out in the middle of nowhere. There was nothing for it to stop it, so I don't know what it was doing. It just thought the tarp was a fuzzy place to pass away. Great. Now, this may not mean much to orchardists, but maybe people who in Africa who experienced giant outbreaks of locusts, that might be something a little more interesting. That's not so much to us. It's just interesting to know. On this graph, we're talking about other beetles. These are non-beneficial beetles, or beetles that just belong to a group I'm not very familiar with. In this case, it was mostly weevils. A lot of weevils. So look at a tarp and see all these long snouted things looking at me. But they were dead. The deltamethrin, again, killed more than the control. There wasn't a difference between the pearl and the deltamethrin this year, showing that even without the insecticide netting, the netting by itself is still preventing them from going anywhere. And I don't know if they're just sitting on the netting until they desiccate and fall onto the tarp, or what's happening there, but something is keeping them there until they die, which is not insecticides. Or hemiptera, or true bugs, it's a lot of these little plant-eating bugs, although that one's a predator, which is beneficial. Same story, that the deltamethrin killed the most. It wasn't significantly different than the pearl, although numerically higher, and the control didn't kill any, which is expected. So far for non-targets, deltamethrin's killing the most, pearl's killing a little, control isn't killing anything. And when we get to the beneficial insects, that really holds true in the fact that the deltamethrin kills significantly more than either treatment. So these are some of the beneficials we found. This one here is called the snakefly. They're a fairly rare insect in the world, but very common here in Washington, which is kind of neat. They can move their heads around, kind of like a snake. They are predatory on aphids and other things. This here is a spider wasp, which has a stink bug in its legs there. It parasitizes it, so it's very beneficial. It kills the pest that we're looking at. Here's some of our native pollinators. So honeybees are not the only pollinator, and they will probably not be as affected as our native pollinators because our native ones live out in that vegetation, and then they fly into the orchard. So you put a barrier up, you're stopping them potentially killing them. This here's a parasitic fly. This guy's pretty cool. He flies up behind a stink bug, lays an egg on its, basically on the nape of its neck. Stink bug lives the rest of its life. Egg hatches, it crawls in, eats the stink bug, kills it, prevents it from producing future generations. Everyone knows about ladybugs and how beneficial they are for aphids and things like that. This here's that syrphid fly, or a syrphid fly, which is similar to the syrphids we were looking at in the cages, which caused outbreaks of aphids when you prevented them. So knocking out syrphids is also not good. There's kind of the majority of the beneficials that we're finding were being prevented by the netting and killed by the netting. But enough of the doom and gloom. What about the good stuff? Luckily, we were killing a lot of stink bugs too. A lot more in the delta metron than in the pearl or the control, showing that even though we couldn't find a difference in the orchard through Tarp samples, we were intercepting and still killing a lot of stink bugs, showing basically these net barriers do have potential. They might need to be tested on a larger scale or on an orchard that isn't commercially managed. And through all of this study, we basically discovered that stink bugs migrated multiple times throughout the year, so that one treatment in August probably isn't going to cut it because they're coming in a lot early. Luckily, they are flying at roughly the height of the vegetation, which is what we expected, so physical barrier is a possibility. All of ours were not huge. They only covered a small portion of that orchard border, maybe bordering up the whole edge might give different results. However, that native habitat is home to a lot of beneficial insects. This is just a side picture of one of my sticky traps after a week. You can see all the things we caught trying to fly into that orchard in just a little 6-foot width. So if you border up the whole border, who knows what all you're knocking out in that area. This was a fun angry praying mantis that I found. If you've ever wondered what those little dots are on its arms, it still looks like ice. So when it flares out, it looks like bigger than it is. And with that, I'd like to thank Dr. Beers and her entire lab crew, and Bruce especially, for helping me build all these crazy structures and dig through rock. I don't know if any of you have ever dug in Manson, but it is literally just rock. And all my funding sources for helping me basically purchase all this netting and these structures and traps. And thank you guys for listening to this talk. With that, I'll take any questions. Yes.
What's the chance that there was diagonal movement of stink bugs from the control into?
Yeah, I would say so. The question was basically what's the chance that the stink bugs moved from the area that didn't have a barrier into the area that had a barrier? And I would say very high, because stink bugs are very mobile. They're a landscape level pest because they move all around. That's why I think if this test was to ever be redone, it should be a bigger section, but it's expensive and it's hard. The big issue with working with people in Manson is that they have hills all over the place, and so building a border barrier that's straight and actually feasible is very difficult. They also know where their stink bug pressure is, and so they kind of like nudge you towards that area to be like, you should put your barrier here. But I would say very likely. Yes, Tobin?
So I was wondering why in your in the smaller cage experiments and then the large cage, you had the aphelinus move in, which are they're tiny, so they would get in through the netting, and then some of the general predators like the lacers couldn't get through, but the earwigs you didn't have any effect. So I'm just wondering, do you think that the earwigs are just maintaining their population within those cages, or do you think they're actually like able to get underneath the cages, or what's going on there?
That's a great question. So earwigs are not a very mobile insect. They kind of stay wherever they're at. I think Robert either cited or showed that they do not move very far from where you release them. And so I'm guessing that those earwigs were there before we built the cages, and that the cages just didn't enter their life cycle that much.
Would have a lot of food.
Yeah, I was gonna say there's so many aphids. But they.
What's the duration of a stink bug flight? So you said, I mean, they'll fly in a straight line, but I mean, there's got to be like a limit to how far it goes.
That's true. I don't know for our natives what the limit of variability is. I know they've simulated flights with the invasive brown marble stink bug by basically attaching it to a wire and saying how long it can fly, and it was somewhere around 40 kilometers. It's pretty far.
Is it one flight?
Yeah, just kept going. But granted, in that case, it's stressed and scared. Actually, I don't know if that's scared. It's stressed. I'd be scared. So that is not its natural flight. I would say normally they're just flying to one of the nearest plant stations. They don't just take off into the wind, usually. Moving again.
So based on this, like, I mean, what are the best possibilities for control? Like, I mean, so given all this information, it seems like it's a little, like, it hints at what might be effective, but there's a lot of other considerations, like, what do you think is gonna be, what are the next steps, I guess?
Yeah. Next steps, I think, would be testing a bigger barrier, maybe without insecticide, I think, because it still blocked a lot of the stink bugs, or having small portions with insecticide netting with those lures attached to it, so it's only attracting the stink bugs and not the beneficials. That would be things I would want to research. I think we need to keep capitalizing on its behavior of migrating and aggregating orchard borders. I do like so far that they basically reduced by 90% the amount of sprays going in to spring borders.
Are there lures in the insecticidal netting? Is that expensive, or is that?
I guess it depends. I'm sure not in the US, it's not very expensive. I think the lures go for between $3 and $10 a piece, but they're effective for basically the whole growing season. The insecticidal netting is effective for, the insecticide netting original purpose was for mosquitoes in Africa. And so it is very resilient to light, and it is long lasting. It's over 3 years in Africa. They're still figuring out how long it lasts here. So the netting, I think, is much more long term. Any questions? Yes.
So overall, where tree fruit is produced, where there isn't a lot of native vegetation, is the relative damage caused by stink bugs negligible?
Yeah, so stink bugs seem to be kind of a spotty or sporadic insect. Down here in Wenatchee, they don't really have any damage from stink bugs. We don't have bitterbrush down here. We have sagebrush, but they don't live on that as much. So it is very tied to the native vegetation.
So ultimately, the best control is to not grow apples near
Right.
native vegetation.
Some people have gone as far as burning the edges of their native vegetation. I think that's a bad thing to do because it provides a lot of other benefits besides just growing stink bugs.
Sometimes it happens whether you want it to or
Right,
not.
yeah.
The burning of the
True.
native vegetation is.
True, that's true. I know during my PhD, the stink bug populations have been a lot lower than when I started working here for my PhD runner. When we would have set out traps, we'd find 100 per trap. Now I'm lucky if I find 5 or 6 per trap, and I don't know if that's just because of life cycles, or maybe we've had a lot of massive fires since then, and it could have knocked out a lot of their habitat.
Or it may have been all of Christian Krupke's research and drove them away.
Yeah,
Yeah.
could have been true.
Just caught them all.
Just got them all. Yeah, mass trapping them. Thank you all so much.
Bye.